Hi, Dr. Exford. My name is Alex. The mnemonic is that Alex is also your name, so I think you already know my name. I had protein kinase A, which is this beautiful heterotetradimer. You can tell it's got different groups. It's got four of them, hence hetero, tetra. Um, let's. I tried a recording before. I thought it was 5 minutes. I was actually talking for 12, so I'm going to be a little bit faster. First, we'll go to question one, which is a buried alpha helix. First off, what defines an alpha helix? An alpha helix is a right-handed helix with hydrogen bonding every 3.6 base pairs from the hydrogen on the amino group of the amino acid backbone to the carboxylic acid of another residue further up the chain. So we can see on this one, sorry, I was moving the thing around. On this one, where my cursor is, um, we can see that this is an internal, like a buried alpha helix, which means that it's not interacting very much with the surrounding solution. And that just means that the groups are more likely to be relatively nonpolar and uncharged all the way around. So as we can see, this one is a tryptophan residue, which is a large nonpolar group. Here we have leucine, another nonpolar group. And then we'll move to just a moment, a surface, a surface alpha helix. We'll move to this one down here because I like green. And this one's going to have some polar residues on the outside, and inwardly facing are going to be mostly nonpolar ones. So you can see we have glutamic acid facing the outside here. We have lysine, which has that um, primary amine group on the end facing the solution because the solution is water. Water is polar. We have on the inside here a leucine, which is nonpolar, and then like alanine, lysine. Well, lysine's a little bit polar, but it's not going to be perfect. And you get the gist of it. So that was two. Three talks about left-handed helices. In this structure, there are no left-handed helices. That's because they're not sterically favorable. The only time you really see left-handed helices are when you have like a specific loop or a specific turn, or you have like a ton of glycines in the structure because they're super flexible and they allow for that kind of steric movement. But in protein kinase A, we really don't see that. Let's move on to number four, which is a parallel beta sheet. Whoop-de-doo, this molecule does not have a parallel beta sheet. It only has anti-parallel beta sheets. What is a beta sheet, you're asking? Well, a beta sheet is a secondary structure just like an alpha helix. Oh yeah, alpha helix is a secondary structure. Primary structure is the backbone sequence. Secondary, alpha helix or beta sheet. Tertiary are each of these groups here, like orange would be a tertiary, and then quaternary is the whole thing. So let's look at we have no parallel. Parallel would mean that the arrows are facing in the same direction. We don't see that. We only see anti-parallel beta sheets on this entire molecule. And let's take you in for a closer look on one of them. Okay, here we go. Wow, that was terrible. Okay, so these two are anti-parallel. Well, I'll talk about parallel first. Parallel basically means they're pointed in the same direction. It's not as favorable because the hydrogen bonds are sort of offset, not at 180 degrees. It makes it so that the formation of these beta sheets is less common. The anti-parallel ones, however, allow for that conformation where the hydrogen bonds are at 180 degrees to the backbone, and we know that structure dictates function, so they're much more likely to actually hydrogen bond. And the spots for hydrogen bonding are the same as that alpha helix. It's from the amino group to the carboxyl. And those were four and five. Um, okay, 10 seconds. I believe I talked about five of them, even though some of them weren't included. I still know my stuff. So yeah, we're coming up on 5 minutes. So thank you, Dr. Rexford. Have mercy on my grade. Have a great night.